Mhm.
Okay, could somebody please confirm that you can hear me?
We can hear you.
Great. We're on chapter 8, but let me check here. Let me share my screen. All right, today is February 23rd, and we are supposed to be starting chapter 14. I don't remember if we got started on chapter 14 or not last time.
We
In the
did. We got
lab,
about 4 slides in.
well, thank you. In the lab, there is no lab today. You're supposed to be working on your unknown project. I will log in at the regular time. Is that 6:30? Let me check. I'll log in at 6:30 to 6:45 to answer any questions. If there's no questions, I'll log off at 6:45. So if you have questions, please log in between 6:30 and 6:45, and I'll answer your questions. There are a number of people who have not started the unknown project, and you probably don't have enough time now to finish the project. But if you have questions about it, ask me, and at least you can get started on the project. If you're very lucky, you might even finish it. Anything else I need to talk about? There will be a quiz next week. We'll talk about that on Tuesday. I think that's about it. Any questions? All right, if there's no questions, let's get started with the lesson. Let's see if I have two here. Yeah, we talked about 4 slides, exactly as you stated. So we talked about the normal microbiota and the host, and how all animals and plants have microbes living with them. And the normal microbiota is only found in certain places, obviously on the surface of people, in their mucous membranes. They're not normally found internal to the body, such as the internal organs and tissues. The human normal microbiota found in all of the mucous membranes, that have an opening to the outside, like the mouth and down the gastrointestinal tract, all the way down to the anus. And actually, the normal microbiota in the intestine is very helpful for us in digesting our food. The normal microbiota is on the skin, in the eyes, and the mouth. The normal microbiota is something that normally colonizes us for our entire life. It might not entirely be that, but the idea is extremely long time. And usually when we get normal microbiota like Streptococcus epidermidis when we're born, we have it until the day we die. There is a transient microbiota, and this might be a microbe that is on our skin for a few days, weeks, or months. And unlike the normal microbiota, it goes away. Once I had Staphylococcus aureus on my skin. I was testing my skin at the time in the microbiology lab. So every 3 months I was testing it. And I went to see my aunt, and she had a Staphylococcus infection skin infection, and she insisted on hugging me, even though she was being treated for Staphylococcus aureus. And immediately afterwards, I tested my skin. That just coincidence. I wasn't testing it because I was exposed to her. I did it just because I did it every 3 months, and it just happened to be right after she hugged me. And sure enough, I had Staphylococcus aureus on my skin. And I said, geez, that's the first time I've gotten positive for that. And fortunately, it was a transient microbiota. The next semester or next term, when I tested my skin, it was gone. So it was something that was on my skin that I picked up from her, and it was just a transient microbiota. Any questions about any of that? And I'm fortunate that was transient, because if you don't know, Staphylococcus aureus is a human pathogen when it grows on our skin. It doesn't always cause problems, but frequently it does. And if you're a patient who frequently gets skin boils, you might have your skin tested to see if you are a person who has Staphylococcus aureus on your skin. Actually, about 20% of Americans do have Staphylococcus aureus on their skin. Most of the time, it's not causing problems, but it, like I said, if you're a patient who frequently has skin boils, that might be because you have Staphylococcus aureus on your skin. What you can do to get rid of it will be very difficult, so I'm not going to even attempt to do that. When we're talking about the normal microbiota and its relationship with the host, you should realize that humans have 1 times 10 to the 13th human cells in their body. And we have 1 times 10 to the 14th bacterial cells living in and on us as part of our normal microbiota. So this begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much, because the answer is, yeah, we are more bacterial, more microbe than we are human. That's partly because microbes are much smaller cells than human cells. Any question about that? If you weigh all the microbes on your person, it would be several pounds. I'm talking about something like 3 to 5 pounds. And that's probably not including all the microbes in your gut. If you don't know, when you have a fresh feces, about half of the weight is from the microbes. Here are some pictures of the normal microbiota. Bacteria in the nasal epithelium, the orange cocci. Bacteria lining the stomach. These are, well, I think they're rods, but they could be spirals. I always thought they were rods, but that's definitely a bent, unless there's 2 of them there. And then here's some bacteria in the large intestine. You can see both rods and coccobacillus or cocci. Many microbes in the large intestine. We found microbiota in some surprising places fairly recently. This is not so recent, 2014, so about 15 years ago now. And they thought that the bladder was normally sterile and that there weren't microbes in it. But they've discovered that in female bladders, there is a normal microbiota in the female bladder. And the study did not look at males, so I do not know if there are normal microbiota in the male bladder. But in the females, there is definitely a normal microbiota in the bladder, so that when you urinate and they do a bacterial culture, like I said, some of those organisms could actually be the normal microbiota inside the bladder. Most of them will be the normal microbiota that come from your urethra. They discovered that mother's milk is not sterile, and it's not unsterile from the outside, that mother's milk inside the breast has microbes in it. So the breast tissue inside the breast has normal microbiota, at least in females. Okay. I don't know the answer to males because they didn't look at that, but the male breast is obviously different than the female breast. So it wouldn't be surprising if it's different, but it is a little surprising that the mother's milk is not sterile and has a normal microbiota, and it is internal, but it is secreted external. And everybody knew that mother's milk, when the baby gets it from outside, it will have the microbes from the outside the nipple. So no big deal, but it's interesting that the baby gets a normal microbiota from his own mother's milk. The placenta has a normal microbiota. Why this is surprising is that the placenta is, the female reproductive system is open to the outside. So it's not a surprise to me that the placenta has a normal microbiota, but apparently they never looked before. And about in 2014, they did look and they found that the placenta has a unique microbiota. And if you don't know, the placenta is only around for a short period of time, at least relatively short. And so it's kind of unique that it has a specific normal microbiota, which is not found elsewhere in the human body. Any questions about any of these? All right, the relationship between the normal microbiota and the host is one of microbial antagonism, and that is the two can compete with each other. And that is good. And that, excuse me, the host and the normal microbiota do not compete with each other. The normal microbiota and human pathogens can compete with each other. And that is good because we do not want human pathogens growing on us. And if the normal microbiota can compete with the human pathogen may not get established. And so the normal microbiota can cause microbial antagonism, competition with human pathogens. And that's good for the host. So the normal microbiota can prevent the pathogens from growing by simply occupying the niche the pathogen may occupy. For example, my skin is covered with the normal microbiota, mainly Staphylococcus epidermidis and Micrococcus luteus, but there's other normal microbes on the skin. And if a human pathogen were to fall on it, like Staphylococcus aureus, and that space is taken up by Staphylococcus epidermidis or Micrococcus luteus, the human pathogen cannot get established. There can be competition for resources. So if the normal microbes consume all of the nutrients in that area, then the human pathogen won't have any nutrients to utilize and then to grow. And so the human pathogen will not get established. The normal microbiota can produce acids, both in the urethra and on the skin. Part of the acid in those environments, meaning the skin is slightly acidic, so is the urethra. And that's largely due, not entirely, but largely due to the normal microbiota making it more acidic. It is true, I don't know about the urethra, but in the skin, that we secrete substances which become acidic, but excuse me, but the normal microbiota metabolize those substances and make it even more acidic. And that makes the environment detrimental for many human pathogens. Any question about that? All right. The last thing that the normal microbiota can do to antagonize the human pathogens is that the normal microbiota may produce bacteriocins. Bacteriocins are something similar to antibiotics. They're usually a protein, but they have a very narrow range of what cells they affect. For example, E. coli can secrete a bacteriocin, and the bacteriocin will inhibit only microbes similar to E. coli. So Salmonella and Shigella will be inhibited by the bacteriocin, and it works, like I said, like an antibiotic, preventing growth by killing Salmonella and Shigella, but it would not harm a bacteria, even another gram-negative like Enterococcus, is that the name? Fecalis, which is a very common bacteria in our large intestine. Any question about that? So this bacteriocin would inhibit human pathogens, Salmonella and Shigella, but many of the normal microbes in the gut would not be affected by that bacteriocin secreted by E. coli. And there are other bacteriocins secreted by other normal microbiota. Like I think there's a streptococcus in your mouth that secretes a bacteriocin. I've always wondered why bacteriocins were not used as a treatment for a human infection, because you can use them like an antibiotic. The difference is antibiotics, even if they're narrow in their scope of action, they usually will hit all gram negatives or all gram positives, or things like ampicillin hit both gram negatives and gram positives, in which case it's broad spectrum. But even the narrow spectrum, it takes a whole type, like all the gram negatives. And bacteriocins are very specific. And like I said, I always wondered why they weren't being used as an antibiotic. And then a couple of years ago, I read an article that they were experimenting with bacteriocins to be used as an antibiotic. So at least they are being, experimented with now. And they'd be a very good antibiotic. Like if somebody had a Salmonella infection, you'd want to give them this bacteriocin from E. coli, and then that would only inhibit Salmonella and Shigella and a very similar bacteria. It would not inhibit many of the microbes growing in your gut. And as you know, when you take antibiotics, they normally, oral antibiotics, they normally kill a lot of the microbes in your gut. And that causes side reactions like diarrhea or constipation, which are very common when patients take oral antibiotics. Any question about any of that? All right. So we can also take probiotics. These are live microbes applied to and ingested in the body, like eating yogurt with live microbes in it. This is intended to exert a beneficial effect. And if you don't know, eating yogurt regularly with live microbes in it, and most yogurt does have live microbes in it, reduces the chance that a person will get a yeast infection. Any question about any of that? It's particularly true of women, but it's also true of men. And then another thing is that after people take antibiotics, it used to be that doctors told them you should eat some yogurt with live microbes, and that will repopulate the microbes in your gut. I don't think they're doing that anymore because the population in the gut normally will come back. It just takes a little time. The relationship with the normal microbiota and the host is one of symbiosis. Symbiosis is just where two different organisms or populations live together, side by side. It doesn't have to be in harmony to be symbiosis. There are three types of symbiotic relationships. The normal microbiota and the host are of the types commensalism, where one organism benefits and the other is unaffected. Some of the normal microbiota are this way. An example of a commensalistic relationship is humans and a nematode in our eye. The nematode doesn't do us any harm, and each of our eyes normally has one nematode in it. It's microscopic, and I'm not sure what it eats. It probably eats bacteria that fall into the eye because it doesn't do us any harm. And there's only one in the eye, and I think the reason why there's only one is because there's not enough food for more than one. So that's a commensalistic relationship. It doesn't do us any harm, and it benefits the nematode because it gets a home and presumably gets food. I'm not sure, but I'm guessing the nematode eats bacteria that fall in the eye. There's also mutualism where both organisms benefit, and many of the normal microbiota form a mutualistic relationship with us. Like E. coli in our gut, E. coli gets a home, E. coli gets food. When we eat, E. coli can get that food, but E. coli helps us digest our food, and so it benefits us. I think I'll just leave it there. Any question about any of that? There's also a parasitic relationship where one organism benefits at the expense of the other. All the human pathogens are parasitic. They get a home, they get nutrients from living on us, and then they do harm to us. Like Streptococcus pyogenes lyses our cells, takes the nutrients out of those cells to grow. It gets a home, it gets nutrients from us. It harms us, it harms our cells, gives us strep throat, and we have a hard time fighting that off, and it can go much beyond strep throat. I don't remember all the old diseases before antibiotics, but many of the human diseases like, I think it was scarlet fever, or maybe it was red fever, something like that. And then it would get into the blood, causing bacteremia, and then would go into the heart and cause grow in the heart and cause disease in the heart. And like I said, Streptococcus pyogenes did a whole bunch of horrible diseases to people before antibiotics. Nowadays, because of the use of antibiotics, it's rare that Streptococcus pyogenes goes beyond causing strep throat. Any questions about any of that? Let me go on and say the reason why Streptococcus pyogenes caused all those other horrible diseases before antibiotics was the human body was not very good at throwing off Streptococcus pyogenes. It has a capsule around it, and so the immune system could not phagocytize the cells, and so the humans had a hard time throwing off Streptococcus pyogenes. And any of you who have had strep throat, you probably know that the human body has a very hard time throwing that disease off. And so usually the strep throat just lasts for a long time. You can't throw it off, and it just gets worse and worse. And finally going to the doctors, and they say, oh, you've got strep throat, let's put you on antibiotics. One thing about strep throat is that infection is very common to come back, meaning normally when they give a course of antibiotics, if it doesn't kill it off entirely, your human immune system will kill it off entirely. And strep throat, when it's very low, the human immune system is not good at killing it off because it has a capsule, and so it can come back. I had strep throat about when I was in 7th grade, and it came back something like 5 or 6 times after that. The last time was when I was in college. Fortunately, it never came back after that. But I knew that after I had strep throat and I got a sore throat again, I would go into the doctors and say, you better test me because I've had strep throat before, and I heard that it's more likely it'll come back, and it's come back before. And like I said, it happened 5 or 6 times it came back. And that wasn't in that many different years, because 7th grade to college would be what? 8, 9, 10, 11, 12, 13, 14, 15, 16. So that was like 5 or 6 times in 9 years. All right, the normal microbiota does not normally cause disease, but occasionally it can. An opportunistic microorganism is one that occasionally causes disease. It does not normally cause disease in healthy individuals, but occasionally it can under certain circumstances. Especially when the normal microbiota colonize an area where they're not normally found, it would cause disease. I mentioned this already. Staphylococcus epidermidis is normally on our skin, but if we have a puncture wound that brings Staphylococcus epidermidis below the skin, Staphylococcus epidermidis can grow in the puncture wound, and that would be an opportunistic microorganism, an opportunistic infection. The normal microbiota may cause disease in people with compromised immune systems, such as AIDS patients. The normal microbiota may cause disease in them. Another example would be people who are taking chemotherapy, like cancer patients. Their immune system is weakened, and they can get an opportunistic microorganism. An example of an opportunistic pathogen would be E. coli when it's growing outside of the gut, such as an E. coli infection in the ear causing an earache, or E. coli growing in the lungs causing pneumonia. A Staphylococcus aureus is normally found in the nose and the throat. I think about 50% of Americans have Staphylococcus aureus in their nose and the throat. In the nose and the throat, for some reason, it doesn't tend to cause problems. But if it grows elsewhere in the body, it can cause problems, such as on the skin. Or if it gets into the bloodstream. Any question about any of that? All right, let's talk a little bit more about the etiology of an infectious disease. Koch's postulates is the steps that are used to prove that a given bacteria causes a given disease. Remember, Dr. Koch used these steps to prove that Bacillus anthracis causes the disease anthrax. And this is the first time someone had shown that a microbe caused disease. And this gave rise, well, this was proof that the germ theory was correct. Now that was not the introduction of the germ theory. This was the proof that the germ theory was correct. Let me blow this out. Up, got that in the way. Come on. Koch's postulates are you have to isolate a pathogen from an animal which is sick. You then have to grow up the pathogen in culture and obtain a pure culture of the pathogen. We're not going to talk about how you get the pure culture. We're just going to assume you get a pure culture. And then you inject the pure culture into a healthy organism, which could be a human or it could be a bunny. And then if the bunny becomes sick, that's why the bunny's on its side, the poor bunny. Koch says that's still not enough. You then have to obtain from this sick individual the pathogen. So you take the blood of the individual, if it's growing in the blood, come on, mouse, and then grow it up in tissue culture and show that it's the same organism that you injected into the bunny. And then that's the steps you use in Koch's postulate to show that this organism causes this disease. Any question about any of that? Remember, Koch's postulate is a topic we've talked about 3 times in this class, so you can expect a quiz question on it in the final. There are exceptions to Koch's postulates. Like, what do you do with a microbe which has unique culture requirements and cannot be cultured in vitro? For over 100 years, it's probably been over 120 years now, people have been trying to grow syphilis in tissue culture. Another one is Bartonella henselae, which causes cat scratch fever. They've been trying to grow that in tissue culture as well. And we've never been able to get either of these 2 organisms to grow in tissue culture. They have unique culture requirements, and we can't figure out what they require in order to grow. And so we cannot grow them in tissue culture. If you can't grow it in tissue culture, you can't isolate it, grow it up, and then inject it into a healthy individual and see if the healthy individual becomes sick. What do you do if multiple organisms cause the same disease, signs and symptoms? For example, there's more than one species of virus that causes the human cold. In fact, human colds are caused by 3 families of viruses. So you cannot use Koch's postulate to prove that one virus causes a disease like the cold, because the cold is caused by many viruses. And then what do you do if some species cause different disease conditions? Like, let me see if I can remember this. Salmonella, I think it's typhi, but it could be one of the other salmonellas, causes salmonellosis, but it also causes, I'm drawing a blank now. What is that? Is it typhoid fever? No. Anyone remember? Sorry. There are 2 diseases that salmonella causes, and I am going to have to look that up because I don't remember. What diseases those are. Oh, good. Didn't get there. Why didn't I get there? There are 2 types. There we go. Well, that's not telling me.
Is it typhoid fever?
It was, it's the lady. Is it Mary Typhoid? I'm trying to remember. And I'm not. I'm sorry. I'm breaking up. Oh, here we go. Here. What's the most serious?
That's right. Typhoid Mary is the right term.
Typhoid Mary, thank you. Yeah.
Yeah.
Yeah, that's it. It's typhoid fever and salmonellosis, or the two diseases that salmonella causes. Thank you for your help there. I was drawing a blank there. And what do you do if the bacteria cause two different disease conditions? Koch's postulates, by definition, do not work. And then what do you do if there's moral and ethical conditions for human-only disease? For example, we believe that HIV causes AIDS. Would we ever use Koch's postulates to show that HIV causes AIDS? You have to isolate the virus, inject it into a healthy individual, and then check to see if they develop AIDS. We would never do that for moral and ethical reasons. Okay, so occasionally Koch's postulates has exceptions. And then what do? Anyone have a guess? Like, how do we know that HIV causes AIDS? Nobody has any guess?
Did we test on animals?
No, because HIV won't grow in animals. It can grow in one of the chimpanzees, but it does not cause AIDS or harm the immune system in the chimpanzee. HIV has to be grown in a human in order to get a disease, and except for the chimpanzee, it doesn't grow anywhere else. So you can't do a human example. With syphilis, you could get it to grow in rabbits. I'm not sure it causes disease in rabbits because the virus, or not the virus, the syphilis bacteria doesn't grow very long in rabbits. You might be able to get a few generations of the bacteria to grow in the rabbit, but after that, it stops growing in the rabbit. But we can get it to grow in rabbits, but like I said, I'm not even sure it causes disease in rabbits. What we do is we follow large populations over time doing epidemiological studies. So you study a large number of individuals, some of them by their own lifestyle came down with HIV, meaning they became HIV positive. We then followed them over time and they got AIDS eventually, okay? And so we know that only the individuals who are HIV positive, meaning all the ones that were HIV negative, never developed AIDS. And only those people who were originally negative and then became HIV positive later developed AIDS. So we follow large populations over time, doing large epidemiological studies, and then we can show that a given organism causes a given disease when we cannot use Koch's postulates. Okay? And they did that the same with syphilis, showing that the syphilis bacterium, Treponema pallidum, causes syphilis. They would study people who were negative for that organism and then follow them, and some of them would pick it up, and then they would have symptoms of syphilis later on. Bartonella and Selly, I'm not sure how they did that. They probably just studied children and studied the children who were bitten by the cats, and then noticed that when they have a cat scratch fever, that they have Bartonella and Selly growing in them. Okay. Any questions about any of that? All right. So for classifying infectious diseases, a symptom is a change in the body function that is experienced by a person as a result of a disease. The symptom is subjective. It is not apparent to an observer, meaning there's no way for another individual to know that the patient has a symptom. For example, when a patient has a headache, there's no way to measure that the patient has a headache. Any question about that? So a headache is a symptom. A sign, on the other hand, is a change in the body that can be measured or observed as a result of a disease. So a sign is an objective measurement. For example, when a patient has a temperature of 102 degrees, that is a sign of disease because we can measure the patient's temperature. That's the difference between a sign and a symptom according to our textbook. Now, usually the sign and the symptom are used this way by textbooks, but even clinicians, as well as in the general public, the two terms are used interchangeably. Okay? And that isn't correct, but I'm just going to warn you that many people use these two terms interchangeably. Like I've got a symptom of a cold. I've got a sore throat, which I think that would be a symptom. But I've got a temperature. That would be actually a sign. Okay, any question about any of that? And I will use these two fairly loosely. You can always ask me, is that a sign or a symptom if you want? And then I will think about it and say, actually, that's a sign. Like when you have a temperature, that's a sign. I don't think there's any way to measure that a patient has a sore throat. I suppose you could look inside and see that it's red. You might see that it's inflamed, but you don't really have any way of measuring that the patient has pain from this sore throat. And that's usually what we mean when we say a sore throat. So a sore throat, I think, is a symptom. A syndrome is a specific group of signs and symptoms that characterize a disease, like the signs and symptoms characteristic of AIDS. Any question about any of that? A communicable disease is any disease that is spread from one host to another. Examples would be measles, tuberculosis, and chickenpox. A contagious disease is a disease that is easily spread from one patient to another. An example of a contagious disease would be influenza, COVID-19, and chickenpox. Actually, we could say measles too. Okay, those are all contagious diseases. What is a communicable disease that is not a contagious disease? Anyone have one? Meaning it's a disease that is communicable, can spread from one patient to another, but it doesn't easily spread. Nobody has an example? It's actually right here. Tuberculosis. It's a communicable disease. It can spread, but it doesn't easily spread. It's not contagious. Another very good example of a communicable disease that is not contagious is leprosy. You have to have long-term exposure to a leper patient or the Mycobacterium leprosy to get it. I don't remember how many hours, but I think it's actually in the neighborhood of something like days. And then actually only a few people will get it, like something like 90% of the population will never get leprosy, but it has to grow specifically on about 10% of the population. And I think it's because of the people's genetics, that about 10% of the people are susceptible to coming down with leprosy. Any question about any of that? If I were ever asked you a question, I would take a very clear distinction. a disease that does not easily spread. That is communicable but not contagious. And I'm having a hard time thinking of another disease, one that doesn't spread that easily. But it does spread. A non-communicable disease is a disease that does not normally spread from one patient to another. Example, when somebody gets tetanus, they're not going to spread it to another person normally. And that's assuming they don't, donate an organ, and then there's an organ transplant. I mean, that might be one way to spread tetanus. But like I said, normally tetanus does not spread from one patient to another. Another case would be botulism. That is a non-communicable disease. It does not normally spread from one host to another. Any question about any of that? All right, let's talk about the occurrence of a disease. To understand the full scope of a disease, we need details about its occurrence, its incidence, and its prevalence. The incidence is the fraction of a population that contracts a disease during a specific time. So the incidence is talking about the new cases only. And usually when we're talking about something like the flu, we're talking about new cases. The prevalence, on the other hand, is the fraction of a population having a specific disease at a given time. The prevalence is both the new cases and the old cases. For example, in AIDS in the United States in 2007, the incidence, those who newly came down with AIDS, was about 56,000, but the prevalence was 1,185,000. So the prevalence is both the new cases and the old cases. On something like COVID-19, you usually see the number as the prevalence, the fraction of the population having a specific disease so far in the time. And I think the fraction of the human population in the United States coming down with COVID-19, I think it's about 60% now of people in the United States have come down with COVID-19. And my data might be a year old, so it may be much higher than 60% now. Sporadic disease is a disease that occurs only occasionally in the population, like typhoid fever in the United States, is usually considered a sporadic disease. An endemic disease is a disease that is constantly present in a population. So the common cold would be an endemic disease in the human population. Malaria is endemic to the tropics. It's a disease that's constantly present in the tropics. An epidemic is a disease acquired by many hosts in a given region in a short period of time. Like when we have an epidemic of influenza, I don't think we have that right now, but maybe we do, I don't know. It's where the cases of influenza are significantly greater than influenza normally is. And so it's acquired by many hosts in a given region in a short period of time. A pandemic is an epidemic that is spread worldwide. So if ever it goes beyond more than one continent, we start calling it a pandemic. Like COVID-19, the world is in the COVID-19 pandemic. Any questions about any of that? Now, when you're talking about a rare disease, there's an argument about whether it is sporadic or whether it is endemic. Like when we talk about tuberculosis in the population in the United States, some people say it's sporadic, but it happens every once in a while. Other people say, no, it's always present, especially in the homeless population, so therefore it's endemic. Because there's so few cases, there's an argument about whether it's endemic or sporadic, and it just depends, I guess, on how you're looking at it, and we're not going to get into that argument, okay? Any question about any of that? Another useful way for defining the scope of a disease is to discuss its severity or its duration. An acute disease is a disease where the symptoms develop rapidly, the disease is short-lived, and then the disease goes away. A classic acute disease is the human cold. It hits us very rapidly, develops very rapidly, and then goes away very rapidly. There aren't too many diseases that are short-lived as the human cold. We could even say that the influenza is an acute disease. Although it's much more severe than the cold, the symptoms do develop rapidly, and the symptoms usually go away rapidly. Even if it's a month, that's fairly rapid when we're talking about a long-term disease. And usually the flu goes away in something like 2 weeks. A chronic disease is a disease that develops slowly, persists for long periods of time, and generally gets worse over time. It doesn't mean the patient may not start getting better. I'm just saying that usually with time, the disease starts slowly and then gets worse with time. That's a chronic disease. An example of a chronic disease would be mononucleosis. The disease starts mild, gets worse with time. Patient gets really tired and sick and worn out. And usually they can recover from mononucleosis and they do get better. Hepatitis B is another chronic disease. There are diseases in between an acute and a chronic disease. We're not really going to talk about them because I'm sure you're not familiar with a subacute disease like subacute sclerosing panencephalitis. I've never heard of that until I got it in our textbooks. So the point is there are diseases in between that, and you don't need to know about subacute diseases. When we're talking about disease, it's important to look at the extent of the host involvement. Infections can be classified according to the extent that the host body is affected. A local infection is where the pathogen is limited to a small area, like a skin boil or an abscess of the skin, where just a local region of the skin is affected. A systemic infection, also called a generalized infection, is an infection throughout the body. Like when a patient comes down with measles, the virus can spread throughout the body. Now, a local infection can become a systemic infection, and that we call a focal infection, where the systemic infection began as a local infection. Like when MRSA, it's multi-drug resistant Staphylococcus aureus, starts as a skin infection and then invades the blood, and then can be spread by the blood elsewhere in the body, causing a systemic infection. Any question about any of that? Bacteremia is the presence of bacteria in the blood. This is compared to septicemia, which is the growth of bacteria in the blood. Normally, when a clinician is testing the blood for bacteria, they're only looking at bacteremia because they're not looking at the growth of the bacteria in the blood. But that is septicemia. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. Usually in a viral infection, we'll have viremia. A primary infection is an acute infection that causes the initial illness. Like when a patient comes down with a sore throat, that would be the primary infection. The secondary infection is an opportunistic infection after a primary predisposing infection has weakened the person's body. Like when a child comes down with the cold, the primary infection, they can get an earache. That would be an opportunistic infection in their ear because of their weakened immune system. Anyone got a question about that? A subclinical disease, also called an asymptomatic disease, is a disease where the patient has no noticeable signs or symptoms. So it's an inapparent infection. Some people can actually carry poliovirus, but never get polio. So they never know that they are infected with polio. A better example is most of you are young, if not all of you, in which case you guys can have an inapparent infection of COVID-19 where you will have no noticeable signs or symptoms. And this will happen about 50% of your COVID-19 infections. Because you're young, you'll have no noticeable signs or symptoms, but you will carry the disease-carrying organism. And then you can bring that disease-carrying organism to other patients, like possibly your grandparent. And of course, COVID-19 would be a very serious infection to your grandparents. Any question about any of that? All right. The severity or duration of the disease can be a latent disease. A latent disease is where the disease-causing organism kind of goes dormant, sits around, and does not cause disease in its dormancy. An example of a latent disease is chickenpox. When the child first becomes infected with chickenpox, he'll have signs and symptoms of disease. That's why we call it the chickenpox. And then the virus goes latent. And it can go latent for decades, for many years. And then the virus activates, comes out of latency, and then causes disease again, which will be the shingles. All the time between the shingles and the chickenpox, the patient had the virus, but the virus was latent. Any question about any of that? Another example of a latent disease is herpes simplex virus 1 causing cold sores. We actually most likely got that infection when we were very young because it's very common in the human population. Over 90%, maybe even over 95% of humans, at least in the United States, have herpes simplex virus 1, and they got it from their parents, especially from their mother, but also from the father, and possible from their siblings. And the virus caused the cold sore when we were very young, and then it goes latent, and it can come out causing another cold sore. Any question about any of that? All right. Herd immunity is when many people in a population are immune to the disease. And herd immunity is vital for preventing the spread of an infectious disease throughout a population. Let me blow this up. If there's no immunity in the population, and then somebody, the red individual, becomes sick with the disease, the disease can spread to nearby people who are around the contagious individual. And then most of the population will come down with the disease because there is no immunity. On the other hand, if most of the population is immunized against this disease, when the individual now becomes infected, most of the population are immune to the disease, so only a few individuals in close contact with that contagious individual will become infected, and then the disease is unable to spread to other people because the people nearby these contagious people are immune. And so the disease doesn't spread to like this individual or that individual because it has to go through somebody who's immune, and it's not going to grow in the immune person. Okay. Any question about any of that? Herd immunity is the whole concept behind inoculating a population for a vaccine. So the idea of a vaccine is to create enough people in the population who are immune to the disease so that if the disease occurs, the disease doesn't spread throughout the population. Any question about any of that? The trouble with herd immunity is you need a fairly high percentage of the population to be immune before the disease will not spread. With measles, you need something like 90 to 95% of the population to be immune to measles before the disease will stop spreading. And the trouble is too many parents are not getting their children vaccinated. And so every once in a while we have measles spreading in the United States. And usually it happens on certain years, and then people realize, oh, measles is going around, maybe I should get my child vaccinated to it. And then the disease stops spreading. But like I said, every once in a while we have measles spreading in the United States. Another example is COVID-19, where they estimated, and these are the original COVID-19, so I don't know if this is true for the new strains of COVID-19, you needed at least 85% of the population to be immune to COVID-19 before COVID-19 will stop spreading. And let me tell you that 85% of the population has not gotten the COVID-19 vaccination. And so COVID-19 continues to spread in the United States and probably in most of the world. Of course, the new strains tend to spread even if you got the vaccine. The vaccine only prevents you from getting seriously sick. And I'm not sure if they made a new vaccine on Omicron for COVID-19. I'm not sure if the new strains are resisting the new COVID-19 or not, but certainly the older vaccine on the original COVID-19 no longer protects against the newly emerged COVID-19 strains going around, although it does prevent you from getting seriously ill. Any questions about herd immunity, where many of the immune people in the population are immune so the disease does not spread? Let's talk a little bit about the patterns of disease. Normally, when disease hits, it follows a specific sequence of events associated with the infection by a microbe before subsequent disease occurs. First, the disease has to come out of a reservoir. A reservoir is just a place where we say the disease comes from. We'll talk a little bit more about what the possible reservoirs are in a later slide. I don't know if we'll get to it today, but we'll talk about that in a minute. Once the disease comes out of a reservoir, the pathogen must be transmitted to a susceptible host. The pathogen has to invade the host and multiply within the host. Furthermore, the pathogen has to injure the host. Without injury of the host, the patient won't have signs or symptoms of disease. The illness then ends either when the host immune system destroys the pathogen, or perhaps a vaccination or antibiotics destroys the pathogen. So the pathogen gets destroyed. That's the main point. Or the pathogen kills the host. That's the other way the illness could end. Any question about any of that? There are certain predisposing factors that make a body more susceptible to and therefore affect the occurrence of disease. For example, females have a shorter urethra than males, so females have a higher incidence of a bladder infection than males. There are inherited traits that make a patient more susceptible to getting a disease. Like sickle cell anemia, that gene makes a patient more susceptible to getting other diseases. The climate and the weather can affect the predisposing factors for a disease. I think everybody knows colds and flus, as well as COVID-19, are more likely to happen in the wintertime. And there's actually a very good reason for that. Not only are we closer inside, we shut the windows in our house, and so the air isn't recirculating. And then we spend more time inside, and so there's people coming in, and then they're more likely to spread the virus in the room we're in, and so we're more likely to get it. So that's a possibility. It's also true that the UV light of the sun is very effective at killing viruses that cause colds and flus. And the UV of the summer is much more intense and more likely to kill the virus than the UV light of the winter from the sun. And then the temperature also helps stabilize the virus. So when we pick up, let's say, something like the flu virus on our coat, and then we go outside, the cold temperature is going to help stabilize the virus and allow it to live longer. Whereas in the summer, of course, we wouldn't be wearing our coat in the summer, but if it's on our clothing in the summer, the warm temperature will decrease the length of time the virus will be alive. And so the viruses are more likely to spread in the cold weather. And this is true very much so for the flu, as well as colds, although I don't think colds really are affected too much by the temperature. And then COVID-19. So COVID-19 is much more likely to spread in the wintertime than it is in the summer. That doesn't mean the COVID-19 cannot spread and the flu cannot spread in the summer. It's just that they're less likely to spread because the virus is less likely to remain alive in the summer for a number of reasons. Fatigue makes a person more predisposed to getting an infection. And this is a problem with students because they're studying, not getting enough sleep, partying, they become fatigued, and that weakens their immune system. Age also affects the predisposing factor. I think everybody knows that the older you are, the more at risk you are for getting COVID-19 and the more serious COVID-19 infection is. This is true for most diseases, although, let me see if I can show that. Not seeing it here, but let me go there. There we go. For most diseases, the population getting affected, so let's make a graph here. The population getting affected is the very young. So how do I do that? And then the elderly. So the older you are, the more likely you are to get a disease. And then the younger you are, the more likely you are to get a disease. Most diseases follow this course. And then at the young age, you're much less likely to get a disease. Any question about any of that? So most diseases follow this. COVID-19 is not quite that way. The very, very young have an increased risk, but it drops very rapidly. And then children are very unlikely to get serious COVID-19 disease. I think they're even less likely to get COVID-19 than a young adult. And then it goes up with age. And so it's mainly the elderly that are at risk for getting COVID-19, and it's much worse for them than it is for even newborns. But newborns are at greater risk for getting COVID-19 than, say, a child. Okay? Any question about any of that? And COVID-19 is an unusual disease in that it's mostly the elderly that get affected. Most diseases follow this type of an example, where yes, the elderly are at greater risk, but young are also at greater risk. And even, I don't know, children like 1 or 2 are still at greater risk. Any question about any of that? A lifestyle can affect the predisposing factor. For example, smokers are more likely to get a respiratory disease simply because they smoke. What happens is the smoking damages the mucosa of the oral nasal cavity and respiratory system, and that makes getting respiratory diseases more likely. Chemotherapy can predispose a patient to getting disease. Not only does chemotherapy weaken the patient's immune system, but it can also damage the mucosa and make an infection more likely. All right, let's talk a little bit about the development of a disease. Once the host defenses have been breached, disease will develop, and disease develops following a certain sequence. And this sequence tends to be similar for both acute and chronic diseases. So almost all diseases follow this course, but not all. And then we'll talk a little bit about the exceptions. Normally, a patient is first exposed to the disease-causing organism, and they have no signs or symptoms of disease. The organism is growing in the patient, and we call this period the incubation period. From when the patient is first exposed to the disease-causing organism till the patient has the first signs or symptoms. That's the incubation period, no signs or symptoms of disease. That's followed by the prodromal period, where the patient has mild signs or symptoms. Like if you've ever asked yourself, geez, do I have a sore throat? And you have to think about it. And you say, yeah, I've got a slight sore throat. That's the prodromal period, mild signs or symptoms. That's followed by the period of illness, where this signs and symptoms become most severe. And usually the number of microbes is also most severe in the period of illness. The period of illness is then followed by the period of decline, where the patient starts getting better. A classic example of the start of the period of decline is when the patient's fever breaks. They no longer have a fever. They're now entered the period of decline, and the disease is going to start getting better. The signs or symptoms start decreasing, but the patient still has signs and symptoms. That will be followed by the period of convalescence. The period of convalescence is when the patient has no more signs or symptoms. What happens is that the number of disease-causing organisms drops below a threshold, so they still have the disease-causing organisms in the period of convalescence, but the number of organisms are below the threshold, so the patient has no more signs or symptoms. And then that's it. After the period of convalescence, the patient is completely cured. They no longer have disease. Okay, any questions about any of that? Right. Sometimes a disease has no prodromal period. Like occasionally I'll be in the incubation period, and then all of a sudden I am hit over the head by a sledgehammer. And what happens is I've come down with the flu. Sometimes the flu does that, where there is no prodromal period. You're in the incubation period, and then you skip immediately to the period of illness. Sometimes the disease has no decline period and no period of convalescence. So the disease, it's the period of illness, and then either always gets worse or the patient dies. AIDS is actually an example where the patient dies when the patient is in the period of illness, in which case there is no convalescence or decline period. Any question about any of that? All right, here's a question for you. In what period of illness are you not communicable, where you cannot spread the disease? Is it in the incubation period, the prodromal period, the illness period, the period of decline, or the period of convalescence?
Is it the period of convalescence?
You would think so. Anyone else have a guess? Meaning it's wrong.
Incubation.
Actually, you're both wrong. You have the disease-causing organism at all periods of disease. So you can spread the disease in the incubation period and in the period of convalescence. It's just less likely to happen in the period of convalescence than it would be in the period of illness. Okay? So that was a trick question. I won't ask you anything like that on the final, but you should know that when you have a disease, you have the disease-causing organism at every period of the disease, so you can spread it. My mother would always tell me, oh, I'm getting better now, so I'm not likely to spread it. And I would tell her, Mother, you're talking to a microbiologist. I know you can still spread the disease. Okay? She always hated that I told people that, but she's no longer alive now, so I can say it whenever I want now. You can cause disease at every period of disease. For the common illnesses like the colds and the flus, you are actually most likely to spread that disease if you're an adult during the late incubation period or the prodromal period. This is for colds and flus. Why adults do that and they're less likely to spread it in the period of illness is their immune system is mounting a response, and that's reducing the spread of the disease during the period of illness. In the incubation period and the prodromal period, the immune system has not responded yet. And so it's not acting. And this is something we find in adults, and it's probably something that evolved with humans to make the disease less likely to spread from the adult to the adult's children. Surprisingly, children, especially young children, do not have that. So they can spread it in the incubation period, the prodromal period, and the period of illness at all periods, okay? Their immune system doesn't seem to limit the spread of the disease. And you don't need to know that. But my point is that for some diseases, the most potent time to spread the disease is late in the incubation period or in the prodromal period. All right, any questions about the development of disease? If not, I'm going to end it here. And I will see you in the lab at 6:30 if you have questions. If you do not have questions, you do not need to come to the lab because today it's only going to be questions and answers. Okay, if you're having trouble with the unknown project, show up and ask me questions. Okay. All right, goodbye.